Hi guys, in this second video we're going to be looking a little bit more at the internal structure of the muscle. So we're going to be basically dissecting a muscle apart and going into more and more detail, basically breaking it into smaller and smaller bits until we're almost looking at the individual proteins themselves. So without any further ado, let's start by pulling a muscle apart. So here we can see the entire muscle, and we can see at either end these slightly paler areas. They're representing tendons which are attaching directly to the bone, and those tendons can't be stretched at all, which means that you transmit all of the force from a muscle contraction directly to the bone, really maximizing the strength of that muscle contraction. But if we peel back the top of the muscle, we see a bit of the internal detail, and we're going to really pull this apart and start looking at what makes up these individual muscle fibers. So from the whole muscle, if we pull that open, we can see we've got these bundles of muscle fibers. Okay, and the muscle fibers are these little blue things on the diagram. Now they wouldn't be blue in real life, that's just to ease your understanding here. And these muscle fibers themselves contain even tinier fibers, which if we now have a bit of a closer look at, we'll see are called myofibrils. Now myofibrils are really where everything happens, and this gives you an understanding of what exactly is going on inside the muscle on a protein level. So what you're looking at here is an image of those individual myofibrils, and you can see that they're a very similar kind of colour to the ones on the diagram. But here you can see these individual striations, these little bands of light and dark, and this is why sometimes skeletal muscle tissue is also called striated muscle tissue. And if we zoom in on that, you can see these bands are very, very clearly defined. And in fact, this individual unit, which we're just going to zoom in on even more, is called a sarcomere. And a sarcomere is really an individual unit, a set of proteins that cause your muscles to get shorter, and that's what we're going to look at in a bit more detail on the rest of this video. So if we move this sarcomere up to the top of the screen and fade it into a different colour, we can examine it in more detail. And we can see that it's made up of these alternating bands of proteins. And the thinner lines here are representing a protein called actin. And they're attached to this one single vertical line called a Z line. You've got to remember that these are repeated all the way along the myofibril. So this is just one of those little bands, on those little striations we were looking at earlier on. And next to these we've got these other proteins represented here by the thicker bands, because it is a thicker band of protein actually, and that's attached to the M line, and we call that protein myosin. And myosin and actin interact, and they pull across each other when you contract your muscle, and that really affects these bands of dark and light that you can see at the top of the diagram, and you need to know the names of these to describe how they change. So if we have a look up here at the top, here's a next diagram of a breakdown of those different lighter and darker greys, and this section here, which as you can see is really made up of the actin filaments where they don't overlap with the myosin filaments, that's the I band. Okay, so it's really important to kind of focus on that. The I band is the clear part where those muscle fibers aren't overlapping. And then you've got this middle bit, which is pretty much the entire area where the actin and myosin filaments overlap, as well as that little bit in the middle near to the M line where the myosin filaments aren't overlapping. That's the A band. And then we also need a name for that little bit in the middle where the myosin filaments are not overlapping with anything, and that's called the H zone. And what you'll need to be able to do towards your A-level exam is you need to be able to describe how these bands, these zones change shape as the muscle contracts. So if I move on to the next slide, I've got a little animation that will talk you through exactly what's happening there. So in this diagram we've shown exactly the same information as we had in the last one, but on this one we can actually show you the motion of the muscle as it, one, contracts, and then two, relaxes. Now be aware that contraction is an active process. Contraction is actually where the actin filaments and myosin filaments are pulling themselves over each other, and that uses up ATP, it's energetic. Relaxation, like now, well, that is being controlled by another muscle, the antagonistic muscle to this one, that will be then pulling it back out. So really all that happens is in relaxation is these two proteins let go of each other and are allowed to pull past each other. Now the bit at the top of this image shows you what the sarcomere looks like when that happens, and you can see clearly that the I band and the H zone get much smaller when the muscle contracts. So that's the area in the very centre, that lighter area in the centre, and the lighter bits on either side near to the Z lines. And you can see they get smaller. But the A band stays the same width, and that's because the A band is defined by the limits of the myosin filaments. So it's really important to remember that the I band and the H zone get smaller, but the A band stays the same size. And another thing I want you to try and picture, and we'll see this in the next image, is that this muscle occurs in 3D. So these actin filaments that you can see now moving in and out of the myosin don't just have a myosin filament above and below, but they're surrounded by myosins, and the myosins are surrounded by actins. So if you have a look at the top right of your screen right now, you can see a transverse section through a myofibril, and you can see these purpley, pinky, reddy circles which represent the myosin filaments, and around them these lighter blue ones that represent the actin. And you can see that each actin has got several myosin filaments about it, and each myosin filament's got several actin around it, and that really strengthens the muscle. That means it's not just pulling itself past one of the filament, but these filaments are working together on all sides. So the animated bit down at the bottom left of your screen, you can see what would happen in 3 dimensions as that's actually working. So you've got all these actin filaments working in concert around the myosin filament, which gives you a lot more strength. And again, we've got the relaxing when they're pulled out and the contracting when they're pulled in. So what I really want you to take from this is an idea of what's happening in the actual sarcomere. I hope you have an idea of what a sarcomere is. It's made up of these fibrous proteins, myosin and actin, and how they react when they contract.